record. Okay, can people hear me? I feel like it's interrupting something. So I'm just trying to catch up with the chat that's going on. Hi, how's it going? Good. Okay, let's get ourselves underway here. Someone is connecting. Yeah, someone needs to mute their microphone. Thank you. Okay. Share, just gonna get share my screen, find my file. Okay, great. Oops, there you go. There's the iClicker questions. And yes, I need to turn on my iClicker, don't I? Let me just do that. Okay, iClicker is just initiating and start class. Okay, so iClicker should be initiated if you guys want to log into that. I'll get you guys back on screen again because I've lost you. And okay, and I will arrange stuff so I can see all my different screens and bits and pieces. See what's happening in chat. Will we finish last? I hope so. Yes, I hope we finished last lecture. Don't worry about. I'm going to say this again and again. Don't worry about getting behind in lecture thing. There are some, you know, there's one lecture in our course that you've already done before on 105. So that, in the worst case scenario, is like a sacrificial lecture. Okay, you will, when an exam comes up, you will only get examined on what we have covered in lectures up until that point. Okay, and so, you know, if I'm not worried about it, you needn't be either. Okay, I'm just going to remind folks where we are in the grand scheme of things. So we're here, April 6th. Let me see if I can just find my pointer. I'm trying to find my pointer amongst all of this, but I can't, I'm afraid. Damn it. I'll have to go without my pointer. I can't find it, I'm afraid. Too many controls going. Got so many different controls going on here. I can't find it here. Let's see my settings. Sharing. I can't find my pointer amongst all of this, I'm afraid. Score in the iClicker, is the score in the iClicker accurate? Is that the score affected in our grade? I don't know. I can't see what the score in your iClicker is. It won't be what's reflected in your grade because there's 15% that's for class participation that comes from iClicker, plus all of those pre-class quizzes. It's all going to go into a big mixer, okay? So the fact that you get, you know, 3 out of 3 in one iClicker session doesn't mean you got 100% in everything. If you got 0, it doesn't mean you got 0 in everything. And the iClicker is only a small portion of that 15%. So, you know, it's a small portion of a small portion. So, in answer to that question, I want to point out that we're still on the protostomes. I just want to note that in a lecture that's upcoming, it's the entire second part of the chapter. In the syllabus, it says 31.3 to 31.4 for lecture 18. It's actually the entire lecture. But the big point is obviously you've got this midterm 2 coming up next week, okay, on 4/13. It'll probably be on Blackboard, not Achieve. I'll definitely let you know ahead of time because there are some issues with Achieve. And so it'll probably be the regular Blackboard exams, which I'm guessing you're all familiar with taking similar, multiple choice in the same way. And it will cover lectures 10 to 17, whatever we get up to, okay, at the end of the next lecture. There will be a review sheet or practice test posted on Blackboard. So practice test, don't know, possibly. There will be 56 questions in answer to that, and you'll have 75 minutes, okay. Regarding review sheets, at the end of each of my lectures, I give quite an extensive summary section. Consider that your revision notes, okay? When you're doing through with that, it's either 1, 2, or sometimes 3 slides of summary notes, okay? Sorry, I'm trying to look at the other questions here. Because Achieve gives no practice questions, you do a pre-quiz. You do 20 questions before every lecture, right? Is it going to be on Blackboard? Probably Blackboard, most likely. I'll definitely let you know, okay? How many questions on the exam? 56 with 75 minutes. My screen is frozen. Try refreshing your own screen and I'll refresh mine as well, okay? Based off the slides. Okay, Dr. Spagnolo's off the slides. Everything that we test on, if it's covered in a lecture, is good for a test. If it's not, then it's not in the test. Will we be able to go back to questions? No. Sorry. Okay, there's a good reason for that, right? Why not? Because I know that you all have WhatsApp, right? So it's an exam.
He's reconnecting, it says.
Sorry, I just refreshed my screen there, okay? If people can hear me again. Because yes. Some people, I don't know if it was my screen or other people's were having issues, okay? I will reshare. Share screen. Okay, share, here we go. Okay, so midterm 2 coming up, that's the important point. I just want to point out, not everybody is doing the pre-lecture quizzes, and this is missing out on easy points, right? So what I'm showing in this graph here is, think of this along the bottom axis, and I still can't find my pointer, I'm sorry, so I can't use my laser thing, because I've got too many screens going on here. But think of this along the bottom axis as like student number, and on the vertical axis, it's the number of pre-lecture quizzes that people have missed so far. And you see that there's a small number of people, 3 people, who have actually missed every single pre-lecture quiz, okay? These make up 15% of your final grade, okay? So you're missing these really easy marks. More than half the class have missed none of them, so that's great. But there's this big slope in between where, occasionally somebody misses one quiz or something, that's no big deal. But, there are a number of students who are missing, maybe 10 or 20 students who are missing a large proportion of pre-lecture quizzes, okay? So don't miss those out if you want to get easy marks, okay? What is the group chat? Okay. So just to let you know that on my pre-lecture quizzes, the settings that I've had, yes, Hassam, you had a question. Welcome back. Okay, yes.
Well, I'll wait until you finish what you said, and then I'll ask my question. I think you might answer it.
Okay. Lowest 2, I think, for grouping. Yes, there will be some that are dropped, like your worst performances, probably the worst 2 will be dropped, okay, certainly on the iClickers. and likely with the class ones as well. The settings that I've done on my pre-lecture quizzes are as follows. You have unlimited attempts, but every time you retake a question, you drop 10%. You lose 10%. So if you got it wrong the first time and you got it right the second time, then you get 0.9 of that 1 mark. Okay. I've made it the same as Dr. Spagnolo, so it's due at midnight the day before lecture, but unlike him, I've actually let you can still go in and do it on the day of the lecture up until midnight, but you lose automatically. There's a 50% penalty on that, okay? So you can still do it late, you get, most of the marks or half the marks. And I'm trying to do them so they appear online, like on a Thursday, 3 p.m., the 2 for the next week should appear, okay? However, just take a note that because we've got midterm coming up and some other reasons, there is none for our next lecture, so for Thursdays, okay? You show up and cannot practice it again. Yeah, but the answers you put show up, okay. Okay, so pre-lecture quizzes, don't miss easy points. This is just a summary of like marks that people have got so far. So here on the horizontal axis, I put the pre-lecture quizzes, the chapter numbers. I've also included the midterm 1 marks on the right-hand side. On the vertical axis is the percent points, and each actual circle represents a single person, right? So you can see that for most pre-lecture quizzes, most people get most of the marks, right? So these are easy marks that that you should do. I don't know what's special about chapter 26, but that seemed to be a bit different. With midterm 1, there's a big spread. The average was about 65% in that, but quite a wide range. This is pre-curving, assuming any curving goes on whatsoever, okay? When is the second midterm? Tuesday, a week from today, okay? Yeah. So, yeah, 4/13, exactly, as said in the chat there. Great. Okay, and I will send an email about it as well as a reminder and let you know the details. Okay, going to move on now and remind ourselves where we were in the grand scheme of our lectures, right? Is that good, though the average just barely passes? It's, that's typical of this class, okay? And it's pre-curving, assuming any curving goes on. Do we have to join the class lecture on the exam? I don't know, but I'll send out, I don't know what you mean by that, but I'll send out the details of how to find the exam on Blackboard. Will the test possibly be curved? Possibly, yes. That all gets evaluated at the end of the semester.
Professor,
okay.
Camilla
Yes.
was asking, like, while we're taking the test, do we have to be in attendance on Blackboard Ultra? Do we have to be on the call while we're taking the test?
No, you just enter the test separately, okay?
Okay.
And I'll post on that. You don't need to be on Blackboard and on the video like we are now. Okay, great. So last time we were talking about the protostome. So we were talking about animals, and we were talking about the Lophotrochozoans. So there's this group enclosed in the black here. I've expanded it out to include all of the protostomes. These are the animals, if you remember, that are distinguished by the fact that when they develop embryonically, the little gastropore that comes in at the bottom of the embryo develops into the mouth first, okay, protostome. And that's true for all of them except actually the arrowworms, which are a bit weird. Okay. And so we've been talking about the protostomes, and we've been talking about the Lophotrochozoans, one of these major groups highlighted in the red box there. Okay. So recap on some of the major points before we move on to look at individual Lophotrochozoan groups. The blastopore that develops into the mouth, except in the arrowworms, as I mentioned before. They have an anterior brain that surrounds the entrance of the digestive tract, and a ventral nervous system with paired or fused longitudinal nerve cords. So the nervous system goes down the ventral side of the animal as if it's going down our stomach as opposed to down our back. Okay. Two major groups of protostomes, the lophotrochozoans and the ecdysozoans, which is the next lecture. And the lophotrochozoans are characterized by a lophophore, a ciliated feeding structure that we talked about in the last lecture. It's characterized by that, but it's not present in all groups, okay? And a trochophore, a ciliated larval form, again, not necessarily present in all of the groups, but typical of many lophotrochozoans. So the groups that we're going to talk about are shown in this phylogeny here on the top right. So we're going to go through these different groups of lophotrochozoans. Just want to highlight some of the groups that have got the most species, because a number of them are sort of small groups with relatively few species, sometimes fairly obscure animals that many people aren't that familiar with. But the major groups include the flatworms, so 30,000 described species approximately. Annelids, so polychaete worms, and another group, the clitellates, that include things like earthworms, 19,000 species. And then the biggest group is this group of the mollusks. That includes things like clams and oysters and snails and octopuses and things like that, okay? So we're going to go through these different groups. And this is the slide that I was on last time, and there was somebody pointed out that I'd made a mistake in this slide. I'd switched around in the diagrams the entoproct and the bryozoans. I've corrected that version that's on Blackboard now, okay? So the group, we're talking about the bryozoans and the entoprocs. This is a clade on the top right of that phylogeny. And the bryozoans are referred to as moss animals. There's about 5,500 species, and the entoprocs, far fewer of them, 170 species approximately, and that translates approximately as anus inside, and that's because they have this lophophore, as in the group the lophotrochozoans, this feeding structure, and we watched a video of that being used in a bryozoan last time. And in the bryozoans, the anus opens outside of those tentacles of the lophophore, whereas with the entoprocs, it exits within that ring of tentacles inside of it, okay? There are also some other groups. So the other differences, these tentacles have cilia on them that move small food particles that they filter from the water column to the mouth. And in the case of the bryozoans, the food particles are carried from the tip towards the mouth. Whereas with the entoprocs, it's the other way around. They actually move up to the tips. And I think that's, I'm not quite sure, but I think it's because then they presumably use those tentacles and fold them down into the mouth to feed on the food. The bryozoans also have a coelom. Remember, that's a body cavity, and a true coelom, which means that both the inside and the outside of that cavity is surrounded by mesodermal tissue. Whereas the entoprocs do not have a coelom, they're acoelomate, okay? So these animals are mostly colonial, so single individual splits into lots of individuals, so they form a colony, and they live inside a sort of house secreted as a body wall, and they are sessile. In other words, they do not move. They're typically attached to the substrate in some way, a hard substrate, although there are a very few number of motile or movable species, and they occur in marine areas, and sometimes brackish and a few freshwater, but they're typically associated with marine habitats. And a great way of seeing them is if you go down to the seashore and find a rocky area that has seaweed, etc. You know, you often see bits of seaweed with these bryozoans living on them. So each one of these little cell-like structures that you can see on this top right diagram is an individual one of those bryozoans, okay? And they're forming a colony all attached there. Can you disperse the larval stages, right? They can disperse the larval stages, right? Even though they're adult sessile sponges. Yeah, so we'll come down to the reproductive bit here. They have sexual reproduction in answer to that question. And most of them, the majority of them, they release sperm into the water that fertilizes, it'll float around and then fertilize another individual, which will then fertilize its eggs and brood those individuals before then releasing the larvae into the water. And they'll disperse around before settling to form a new colony. But there are a few entoprocs that actually release their eggs rather than brooding themselves before releasing the larval stages, okay? And as I mentioned, they're common in rocky coastal areas. It's a good place to see them if you go down the seaside. Okay, so we'll move on to the next group of these, which is, so here within the Lophotrochozoans, which are part of the protostome group. And in this top sort of major clade here that the bryozoans and entoprocs are part of, there's also these other three groups, the flatworms, the rotifers, and gastrotrix, okay? The flatworms have more species than these other groups. I put up this table last time that just shows us actually quite a wide variety of body forms amongst the Lophotrochozoans and the protostomes in general, okay? That has, before genetic methods, made their phylogeny very difficult and convoluted and incorrect in many cases, because people use a coelom structure basically as one way of classifying animals. That doesn't work correctly when you look at the genetic sequences. So amongst this group of flatworms, rotifers, and gastrotrix, even though they're relatively closely related, they differ quite a bit in terms of flatworms have no coelom. They've lost a coelom effectively. Rotifers and gastrotrix have a pseudocoelom. And then they also differ in that the flatworms have a blind gut. Okay, we talked about that a bit last time, a blind gut being where you have a gut, but there's only a single entrance, okay, or exit, okay, as opposed to a separate mouth and a separate anus from that. Whereas the rotifers have a complete gut. They have, an opening at one end and another end, okay. So let's talk about the flatworms first of all. These are also, they're called platyhelminths, that's the name of the group. And so we just talked about how they have no circulation system, but they can nevertheless become, a reasonable size with these things. And because they're so flat, despite the fact that they have no circulation system, their flatness gives them a large surface area and means that their tissue is always close to the surface. So you can have diffusion across the surface for things like gas exchange, etc. So dorsoventrally flattened, so top and flattened. Quite a large number of species, 30,000 described species. Quite a large number of these are free living, but the majority of them are actually parasites. In other words, they're not free living, they're living on or inside some other animal. Amongst the free living forms, they have a mouth opening to a blind gut, so no anus, so that's the blind gut that we talked about. And they have a gut that has is highly branched. So remember, they got no circulation system. So that highly branched gut means that that there's good diffusion, good surface area within the gut for diffusion to tissues. Some of them are cephalized. In other words, that means they sort of something as a recognizable head, as shown in this bottom right diagram of a free living flatworm. You can see on the left-hand side of it, there's what looks like a head end to it that has got eyes and chemosensory organ, so there's a direction to that, to the animal. And they glide on mucus using cilia, these free-living forms. And most of them fall within this large group called the Turbellaria, okay? About 4,500 species. However, I was saying that the majority of flatworms are parasitic. So are they basically composed of insects? Can they disperse larvae? I don't understand that question. Composed of insects. They're not insects. Okay, sorry. They're definitely not insects, okay? They're flatworms. That's okay. There's no dumb questions, right? There's no dumb questions. And, you know, there are so many different groups that we go through. It can get, I mean, I get lost myself sometimes. It's good to zoom out and remember where we are in the grand scheme of things sometimes, right, before you zoom back in again. Okay, that's why I like to put these phylogeny trees up at the top right that they have in the book as well, because it's a good reminder of where we are in the grand scheme of what we're talking about, right? So there's two important endoparasite groups. One of them is called the digenians, and these are typically referred to as flukes. 11,000 species, and they have a complex life cycle. We talked previously about how quite a large number of parasites have these complex life cycles where, you know, they'll parasitize one species and then be released or eaten, and then parasite some, you know, completely different other organism, etc. So complex life cycle. In many cases, that includes a life cycle that's going through a land vertebrate and an aquatic snail. And some of these are actually really important human diseases. So, for example, bilharzia. If you're in a sort of tropical place, like many places in Africa, for example, if you go swimming in still waters there, in like ponds and lakes, for example, getting this bilharzia disease is a real risk, and snails within that system are sort of like completing that life cycle. Another group, important group of parasitic flatworms are the cestodes or tapeworms, okay? Try refreshing the page, refresh. Okay, somebody's having problems. Is it just me, or does anyone else not hear refresh? Okay, so I think it's just you. Everybody else can hear me, it seems like, right? You're good, Professor. Okay, thank you. But you can still hear me, right? Tapeworms, we actually showed this example before. This bottom right picture is a life cycle of a cestode that I used in a previous lecture that goes through stickleback fish, which is then eaten by a bird, that then in the feces, the eggs come out and hatch and go into a copepod that's then eaten by the fish. So this is an example of a cestode or a tapeworm. Again, there's you know, plenty of forms that infect humans as well. Can be very long in length, and they have no mouth or gut. They absorb nutrients through the skin. So these things are actually living inside a host, like inside your intestines, for example, or in the case of the stickleback, inside its peritoneal cavity. And so they're bathed in this fluid that then they're just absorbing nutrients directly across their skin, so they don't need that gut. And then the other major group of parasites in the flatworms are the monogenes. So these are external parasites that typically have a direct life cycle. In other words, a simple life cycle where they live on a single host and can reproduce and then directly infect that same host species again. They don't have to go through lots and lots of different complex species, okay? These are important parasites of fish in particular. They usually live on the skin or gills or fins of fish, but also some other aquatic animals as well. And they can be a real problem in aquaculture and things like that. If you get monogene infections on gills, it stops them being able to breathe properly and suffocates them. So in a general sense, this last point here is it's hypothesized by some people that the way that this transition to parasites in general has occurred in flatworms is that there were free living forms that were living on dead animal material. And then that transitioned into the fact that that form could also then feed on the surface of, rather than being dead, an organism that was dying. So that would give a transition towards something that would be an ectoparasite. And then from beyond that, there could be a transition towards forms that were endoparasites living inside a host. So that's a sort of hypothetical transition about how those different parasitic forms might have evolved. Okay, so we talked about the flatworms, okay, but there were these two other sort of like smallish groups of related organisms. And the first of those is rotifers. Okay, so there's about 1,800 species, mostly freshwater and very tiny organisms. So 50 to 500 microns. So that's the biggest size would be 0.5 millimeter. Okay, so it's not something you sort of typically see unless you're going to look at some freshwater under a microscope. One of the remarkable traits of these is that some of them can resist desiccation. So in other words, they can completely dry out. If you have an ephemeral pond that dries up, these also can dry up and remain in a resistant desiccated form, which then come back to life when, you know, maybe it rains again or something like that. So they have a complete gut with a mouth and an anus, as opposed to the flatworms, as we mentioned before, and specialized internal organs. They have a pseudocoel, okay, that means a coelom, a body cavity, surrounded on the outside by mesoderm, but because it's a pseudocoel on the inside, it's surrounded by endoderm, okay, not by mesoderm on the inside. That can act as a hydrostatic skeleton, okay. Remember, that means that you have a fluid-filled sac that if you squeeze it, it's because fluid is non-compressible, it can be used for locomotion, okay. But most of them don't actually use that for locomotion. Most of them are cilia, you know, they use cilia for moving around. And they feed with this specialized organ called a corona. Now that's this circle of cilia around the mouth. Okay, corona, I believe, relates to crown. So that's what that's referring to, the crown of cilia. And that's used for feeding, bringing foods, so filter feeding from microorganisms in the water column. Yes, it's crown in Spanish. Okay, good, thank you. And then that food is mashed up by an organ called the mastax, okay, a sort of complex muscular structure that will grind that food up for digestion. Okay. And the third in this group of related organisms are the gastrotrechs, okay? Oops, sorry, we're still talking about the rotifers here. There's one more slide about the rotifers. Males and females, separate sexes in some species. There's a group called the deloid rotifers that only have females though, and the eggs develop without any fertilization. And the strange thing about that is that you think, well, if there's no sexual reproduction, you probably remember from your previous lectures that that can lead to the accumulation of deleterious mutations. If you're not reshuffling your genome and sort of selecting out the good and the bad, basically. However, and this is apparently perpetuated for millions of years, and so it seems surprising that it's been successful. But it's thought that they can pick up gene fragments from the environment during their desiccation recycling phases. Exactly how that works, I have no idea, to tell you the truth. But it's a mechanism, a genetic mechanism that counteracts the fact that they don't have this, this sexual reproduction part of their cycle. There are also some highly reduced forms descended from free-living forms. Spiny head worms, which are parasites with complex life cycles and several hosts, and another group called the jaw worms, which are tiny marine organisms that live between grains of sand, on the beach or in subtidal areas. Okay, so some minor groups of these. These are actually, the spiny head worms are actually important parasites in some, like fish, for example. Okay, and then are jaw worms harmful? I don't think so. Okay, I think they're no, not to my knowledge. Gastrotrix. So their common name is hairybacks. Again, a relatively minor group, about 800 species. Something that are not typically observed because they're so tiny, only 0.5 to 3 millimeters. Although they're extremely abundant in marine sediments, freshwaters, and soils as well. And they're pretty similar to the rotifers in terms of their general appearance, but they have a flattened ventral surface that's covered with cilia. That distinguishes them from, or it's a trait that distinguishes them from the Gastrotrix. And they're mostly simultaneous hermaphrodites. Okay, so what does that mean? It means you can have male and female organs. That's the hermaphrodite bit. The simultaneous means that you can express both that male and female at the same time. Okay, as opposed to organisms that are, for example, sequentially hermaphrodites, where you're a male first and then a female later on, or vice versa. Okay, these are simultaneous hermaphrodites. But male organisms have been lost or reduced in some species. Okay, we're probably aware of the fact now that pretty much most of the things that we go through have got these exceptions where other types have evolved, basically. Okay, so just a reminder where we are. We're still within the Lophotrochozoans, which is a major group within the protostomes. And we've talked about this top major clade that has the bryozoans, flatworms, Gastrotrix, etc. Now we're going to focus in on the bottom major clade here within these Lophotrochozoans. And the first group we're going to talk about are ribbon worms. Okay, it's about 1,200 species. And these are mostly small, but some may be up to 20 meters long. So it's very variable. And again, this is not an organism that you would typically sort of think of or see or something like that. But if you wanted to see one of these, another good way of, just like with the bryozoans, is to go down to the seaside. And if you start digging in the mud or the sand on a beach in Long Island, it's pretty common to come across these things. They're called milky nematines. Nematines is the name of the group of the ribbon worms. And you'll dig up some sand and you'll see this kind of like sort of piece of thin spaghetti almost, which is what these worms are. And they're common in the seashore if you start digging around. And they're highly predatory. They move between grains of sand or over the surface of the sand, and they feed upon things like clams and things like that. Okay, so these are predators moving around in the intertidal or subtidal area. They have a closed circulatory system and a complete digestive tract. Okay, so a mouth and an anus with a passage all the way through, versus the flatworms. Okay, which if you remember we just talked about, that have a blind end gut, or in some parasites they've lost the gut altogether and no circulation system. Now I mentioned that many of these nematine worms many of them are highly predatory. The way they do that is they have an organ in their mouth called a proboscis, okay, a muscular proboscis that is contained within a fluid-filled cavity, and that basically avert that and can pierce. So it has some sharp stylets, so little sharp pieces on it basically, that can pierce the skin of their prey, and in some situations can release toxins as well. So that's how they go about hunting their prey. So this is showing at the diagram the proboscis when it's not been averted on the top part of the diagram, and then when it's averted by that fluid pressure, it comes out, and then at the end of that is that little tip that can pierce the prey. Okay, we'll move it down to this next group here, the brachiopods and the phoronids. Okay, never going to swim in the shallow. Okay, these things, they're not going to attack you. Okay, Mark, so you'll be in good shape, I think. Okay, probably more likely to watch out for the floating trash or something like that if you go down in Queens, I think. But the brachiopods and phoronids. Okay. Both of these groups use a ciliated lophophore. That's the organ that we talked about last lecture. We showed a bryozoan using a lophophore. It's characteristic of the lophotrochozoans, although not all lophotrochozoans have them. So this is an example of some of the lophotrochozoans that have them, although it's actually believed to have been derived independently from those of the bryozoans and the enteropods. So this picture on the right shows a brachiopod. They're called lamp shells. The one on the left is a phoroniid. So you can see it's got this, the phoroniid has got these tentacles as part of the lophophore that it's ciliated, and is a ciliated feeding organism, so they're feeding upon plankton and microorganisms in the water column. Okay. Brachiopods, there are about 450 species, and it's a group that's really well represented in the fossil record and goes back a long, long way. So this is a group, almost a sort of fossil organism that hasn't changed much in about 400 million years or so. And this is because they fossilize very well, because they're made of these shell-like structures. They're solitary animals, so they're not colonial. They live as individuals, about 4 to 6 centimeters, and they're attached to the substrate by a little flexible stalk that's at the back of the organism there. And superficially, these look very much like, a clam or something, right? They don't seem to be that much different. They got these 2 shells that close like this, but they're entirely different from a clam. And one of one of the reasons is they have this lophophore for feeding. The other is that those shells are dorsoventral, so from the top and the bottom, and they're closing like this. Whereas in a mollusk, they're lateral. It's a left and right valve closing like that. So that's one distinction between the brachiopods and bivalves, the clams and things like that, that are in a completely different group in the mollusks that we'll talk about in a minute. So 2-part shell, and the 2 shell halves are dorsoventral. Okay, yes, we talked about that. Now these phoronids are called horseshoe worms, really minor group. There's only 10 species that live in marine waters, okay, down to about 400 meters deep. 5 to 25 centimeters, and they live inside tubes that are made of chitin, which is different from an animal called a chiton that we'll talk about in a little bit. Okay, it's chitin with an I, and they live in they have a U-shaped gut with an anus that's outside of the lophophore, like the enteroprocs in that respect, as in the anus opens outside of the tentacles. Okay, we're coming down to the annelids, a big group of lophotrochozoans, 19,000 species. Not as speciose as the mollusks, but still quite a lot of species within them. And these are segmented worms, basically. Worms that have these clear segments that we talked about in previous lecture, with a coelom in each segment that is isolated. So we looked at, is there from kind of native to California? Is that species native to California? I don't know, but I'm guessing so. Okay, in answer to that question. So we we looked at how worms move in a previous lecture, and discussed how the fact that because they have these segments, and each segment can act independently, and the coelom is sealed within those, some parts of the worm can expand while the others can, get thicker, etc. That helps them move. Okay, so they have this clear segmentation, and each segment is isolated from the coelom in most of them. They also have a separate nerve ganglion that controls each segment. So each segment is controlled by its own sort of pair of nerves that come off, okay? In this bottom here, it's showing a sort of diagrammatic representation of that nervous system. So remember, these are protostomes. They have a ventral nervous system. It goes down the side of them, and then in each segment you've got nerves that come off that are controlling movement within each individual segment. So annelids. So most have a thin permeable body wall that serves for gas exchange, and they're generally restricted to aquatic and moist habitats, and that's because they have that thin permeable body wall. If they're just on a dry land, they'll dry out basically. And there are two major groups: the polychaetes and the clitellates. Okay, so the two cegans are different from annelids. We'll talk about C. elegans in a minute. Okay, but they're not segmented for a start. Okay, they are worms, but they're not annelid worms, okay, with regard to C. elegans. They are actually in the next nematodes. Yes, exactly. They're nematode worms, roundworms. So yeah, there's a lot of worm-like things, right? So it gets a bit confusing with the different groups. So within the annelids, you have the polychaetes and the clitellates. The polychaetes are shown in these two bottom left diagrams, and examples of the clitellates, the earthworms and the leeches in the other group. Okay. So let's talk about the polychaetes first, and they are see that the polychaete refers to poly meaning many, and chaete, or the chaetae are also known as the setae, like these hair-like structures that they have coming out of each appendage. So they're mostly marine, often in burrows and soft sediment. So if you dig up some marine sediment, there are lots of polychaete worms within these things. It's used in a lot of studies of pollution, etc. in the ocean. People will dig up sediment and look at the different types of worms which are characteristic of pollution levels that you might have. Turns out it's not a single clade, it's a paraphyletic group. So remember, that means it's only part of a group. It doesn't include all members of a clade. Some of them are motile. They're referred to as errant polychaetes that can crawl and burrow through the sediment, and they're often predatory. Like this bottom left-hand one, they move around. Fishermen like to dig them up on the shore for these so-called nereid worms that they use for bait. They're great for bait, basically, for a lot of things. And these mobile ones have what's called parapodia on each segment. So parapodia is like an appendage, like a little foot, basically, that comes out on each side. So in the diagram here, you can see there's an extension from each segment, and then at the tip of those there are the setae, the little hair-like parts that come out of them that can be used for locomotion or other purposes as well, such as like being spiky for defense. There are other types that are sessile, that live in tubes. That's shown by the second diagram. It's stuck within the sediment. It has this this whirl that comes out of the top. It's not a lophophore, and that is used for filter feeding. It is ciliated though. So these tentacles for deposit or filter feeding. So deposit means picking stuff off the surface of the sediment. Some of the most extraordinary polychaetes are a group called the Pogonopherans, which are incredibly specialized group of worms that live in deep water. And what's so special about them is they have some endosymbiotic bacteria, so bacteria that live inside of them that are chemoautotrophs. And so this is extraordinary because these organisms, rather than ultimately making energy and carbon compounds for using sunlight, so fixing carbon using sunlight, they were able to fix carbon by oxidizing sulfur compounds. So there is a group of organisms on the seabed in deep areas that can live without sunlight, basically, which goes against what these are shown in this diagram here. They live in deep sea vents. So these are deep parts of the ocean where there's volcanic activity that is releasing sulfur-rich compounds. Is the light in the photograph just from the camera? Or is this somehow a natural light? No, the light in this photograph is provided by the camera. Yes, this is like way below. This comes from probably, it can be variable, you know, 1 to 4,000 meters, and there's definitely no light there. So these are that sulfur-rich compounds coming up from the hydrothermal vents is ultimately the basis of life for that, in terms of fixing carbon, and then they are predated upon by some other animals, etc. So this is incredibly unusual and quite remarkable. Okay, so the next group, so we talked about the polychaetes. The other major group of annelids are the clitellates, about 3,000 species. That includes things like earthworms, but also leeches as well. So these do not have the parapodia, the little foot structures that stick outside, or tentacles, and only have, if any at all, a few setae, those little hair-like structures coming out of them. They're hermaphroditic, so male and female. So an earthworm, when they mate, they both give sperm and they receive sperm at the same time. And leeches are unusual in that the coelom is not divided in that compartmental basis, and that they have these suckers at each end of them that they can use for attaching, and they act as ectoparasites. So they use those for attaching to a host species and then feeding on the blood from that host. How are we doing in time? Okay, we've got 5 minutes to talk about the mollusks. Okay. So with the mollusks, this is the biggest, most diverse group of the Lophotrochozoans. Okay. And so over 100,000 species of them. And they're divided into 4 major clades, so 4 major phylogenetic groups that include the chitons, that's this top diagram, gastropods, which are snails and things like that, the bivalves, so clams and oysters and the like, not the brachiopods, they're different, and then the cephalopods, octopus, squid, things like that, okay? In a couple of slides, I'll talk about 3 major body compartments that are common to the mollusks. They have a muscular foot, a visceral mass, and a mantle, and I'll describe each of those in a minute. So in most species, the mantle, which is like a sort of protective layer, basically, contains gills that are used for gas exchange. And in many groups or many types for filter feeding as well, particularly in the bivalves. Okay. Also, mollusks have this unique feature of the mollusk called a radula, which is like a tongue-like structure with teeth on it that can be used for rasping algae and food off surfaces, or has been modified into other forms in other groups. There's a video there that I'll let you watch in your own time. It's only 40 seconds that shows an example of that radula in motion. But it's a unique feature of the mollusks. They had blood vessels that do not form a closed loop, except for in the cephalopods. Instead, they have a thing called a hemocoel, which has got some vessels that empty into a common area that bathes the tissues in fluid, which is then collected and then recirculated. Okay, so rather than just being in blood vessels the entire way around the organism. Okay, so I mentioned the 3 body components of the cephalopods. Okay, the first of these is the foot. It's a muscular structure originally used for locomotion and supporting internal organisms. So the schematic next to each of these 4 major groups shows where that foot is. Okay, so the foot of the gastropod is that large part that you can see attached to the leaf, for example. In bivalves, it's a structure that can be used for by clowns for digging into the substrate, for example. And in cephalopods, it's been modified into the arms and tentacles. Okay, so different ways of using it. And in the chitons, it's used for attaching to rocks. Okay, the second is the visceral mass. So the visceral mass is just like a sac that contains all of the major body organs, okay? The heart, the digestive, excretory, and reproductive organs. They're all concentrated in this kind of like little loose sac. So if you break the shell of a snail, for example, and take it out, you'll see there's the foot that you can see there in that picture. But then there's this kind of like loose, thin-walled structure on the top that's got its organs inside of it. That's the visceral mass. And then there's the mantle. Now the mantle is a fold of tissue that basically covers the visceral mass. And it's the piece of tissue that is responsible for secreting the shell that you find in nearly all mollusks. So, for example, in the gastropods, so the black line there shows where the mantle is, and that is secreting the overlying shell. In bivalves, there's these folds that come down each side of the organism that secretes the shell. And in this bottom left picture of a cowrie shell, you can see that this mantle, it can come out and fold around the shell, and it can be retracted again if it's threatened. But that's the mantle that you can see on the right-hand side of that picture of that cowrie shell. Are chitons and bivalves sessile? So I'm just looking at the things here. Chitons, they're attached to rocks, but they can actually move around on the surface of the rock. With regard to bivalves, they can typically move around quite a bit, either by digging through the sediment, or some types of scallops can actually flip along using jet propulsion, and so bivalves can be mobile. Others, like mussels that you see attached on rocks, are actually attached by threads to the rocks, and they are sessile, so it's variable. Okay, great. Once again, we're out of time. Okay, so here's the reason why I'm not worried. You guys in 105 have done developmental biology already, which is listed as, and Dr. Baker often has that within 106. If we lose developmental biology because this is taking time, there's a large content to these chapters, it doesn't matter. Okay. Oh yes, don't wait. Yeah, 1:30. Okay, so we're not out of time. Sorry, I'm getting ahead of the game. But still, thank you for reminding me. I'm getting my times mixed up. But anyway, that's why I'm not worried about time. Okay. Okay, so we still have time to go then. Yeah, I was thinking, holy moly, we haven't got through this lecture in two sessions. So we got time to talk about more about mollusks, and the first group were these chitons. Okay, somebody was asking us, are they mobile or sessile? They are attached to rocks, but they can move around on the surface of a rock. I didn't take 105 because it was full. Okay, you'll probably have to say 105 in the future, though. Okay. Okay. So these chitons, what's characteristic about them is they have 8 calcareous plates that you can see like a suit of armor on the surface of them, right? Attached to rocks. Most are marine omnivores, so they can eat algae as well as animals as well. And they use that radula structure for feeding, okay? Let me just, because I got my timing wrong, let's go back to that video, okay? Here it is here. See if I can get that to play. Okay. So what you're seeing here is this, it's like a little conveyor belt that's got all these little teeth on it. And it comes out and it scrapes. And what's also cool about this radula that's used for a scraping organism is it wears out, but it's continually being renewed. So when you if you take one of these out, you can see that it's got developed little teeth at one end, but at the other end, it's got little developing teeth. What is this? This is the radula, the feeding structure. So this is the mouth of a chiton, actually, it says here. And what they've done with this is that they have attached the chiton instead to a rock, they've attached it to a piece of glass, filming from underneath, okay, to looking, to seeing how that is feeding, okay. So it's like a little conveyor belt they use for scraping the rocks, okay. Okay, so we were down here. Okay, and they cling tightly to rock surfaces with a large muscular foot, one of those 3 parts. Now, if you go down the seashore and look at the rocks, more typically, it depends where you are, basically, you will see this thing in the bottom right-hand corner that is also a mollusk that attaches to the rocks, that feeds in just the same way, basically. But these are actually not chitons, these are limpets. These are modified gastropods, so snails, basically. And you can tell that they're different because they don't have these 8 overlapping calcareous plates, okay? This is just a single shell in the bottom right-hand picture that's stuck to the rock. So 2 similar types of mollusks that are in different groups that have evolved to live in basically the same habitat in the same way. Are the calcareous plates purely for protection, or do they have another function? I believe they're just for protection, to the best of my knowledge, okay? And so, you know, some species of these chitons can actually get really big, and in some places they're long-lived, and they're actually eaten in some places as well. Okay. Second major group of mollusks, okay, the gastropods. So this includes things like snails, so either land snails or marine snails, little snails that you'll see attached to, again, a lot of these things you see on a rocky shore that you would see on a rocky shore. Slugs, those limpets that I've just showed you are modified gastropods. Nudibranchs, so marine nudibranchs shown in this white and blue picture, that have effectively lost their shell altogether. That's why they're nude. And then the bottom pictures of an abalone, okay, popular long-lived mollusk that people eat on the west coast. So nudibranchs and slugs, their shell has been lost. Many of them are are toxic with these warning colorations. And the way that they gain these toxins is, in many cases, it's through the food that they eat. So certain nudibranchs, for example, feed upon sponges that have toxins within them, and then they express those toxins themselves. So they kind of hijack that toxin from their prey. Nudibranchs are really cool, crazy. Exactly, yes. Sorry, just looking at the comments there. There's the other types that actually use, we talked about the cnidarians last time, sea anemones and the corals and those groups that have these stinging cells with the nematocysts that sting. Some types of nudibranchs can actually feed upon cnidaria and take their stinging cells and express those in those little gill-like filaments they got on the dorsal side there, and use those as stingers instead. And these warning colors are to predators that basically say, watch out, I'm toxic, or I'm going to hurt you in some way. So they're mostly aquatic, but some are terrestrial, as we know from seeing slugs and snails in our yard. And these are the only terrestrial mollusks. Land species use the mantle for gas exchange, okay, rather than gills. So the mantle, if you remember, was that bit that goes over the visceral mass that is responsible for secreting the shell. Okay, so we talked about 2 groups of mollusks out of 4. So now the third group we're going to talk about are the bivalves. So these are things like clams. This is a hard clam, Mercenaria, that is the top right one that's popular. You know, you get lots of them in Long Island Sound. And this bottom right picture is oysters, intertidal oyster reef. Again, this is from Greenwich in Connecticut, this oyster reef. And so these are mollusks that have 2-part shells. And as I mentioned before, these are left and right, they're lateral shells, as opposed to those of the brachiopods that we talked about earlier on that have the dorsoventral shells. Okay. And many of them use the foot, so part of that molluscan body feature, for digging into the sediment. So in that top left diagram, you can see the bottom part of it. It's like a tongue coming out of it. That is a very muscular type of foot that they force down into the sediment and then grab on and then pull themselves down. Some types of bivalves that can actually do this amazingly quickly. It's quite impressive to watch. Feeding by bringing water. Yeah, okay. So a lot of bivalves, or the majority of them, are filter feeders. So they have these gill structures that hang down inside the mantle. And those gills, as well as being used for gas exchange, they have cilia on them that are moving water all the time. And that water moves past the gills by coming through a siphon, as you can see, like a snorkel coming up at the top of the sediment in this clam. Water comes down one of the siphons over the gills and then goes back out the other siphon, the exhalant siphon. And as the water passes over the gills, tiny food particles, phytoplankton, sometimes some zooplankton as well, are trapped on the cilia, and those cilia beat and move them to the mouth. So filter feed. Okay, and what's a siphon? So the siphon, if you haven't got my laser pointer, I don't know at your end whether you can see my white arrow that I'm moving around. Yes, you can. Okay, good. So in this bit here, the siphon refers to this little tube coming out the top of the picture. Okay, it's like a little snorkel that's coming out at the top of the sediment, and a tube that goes down inside the mantle cavity where the gills are. Okay. Is it like the lungs? No, it's not like the lungs really. It's just a tube. Gas exchange is occurring in the gills, which are inside the mantle cavity. So the gills are inside the shells, okay? Each shell has a mantle cavity coming down the side, and then there's a hollow area in the middle where the foot is, but also some gills that come down the side. So what it's doing is the siphon is just a tube that's bringing in the water. Then it circulates around the gills, and then it goes out the other siphon, okay? Okay. Okay, last major group is the cephalopods. So these are the squid, cuttlefish, octopuses, and nautilus, each one of those there, okay? These are typically highly mobile, and the way that they move around is that they've modified their mantle cavity, the cavity caused by the mantle, so that they can use it as for jet propulsion. So imagine that siphon that I was showing you with the bivalves. If you could squeeze and then force water through that, it can be used for jet propelling you in one direction or another, okay? They're well known for having well-developed brains and complex sensory neurons and excellent eyesight. We don't have time to talk about it, but their eyes, you know, sort of are actually better designed than ours in certain ways. They have excellent eyes. And their arms and tentacles are used for movement and capturing prey, okay? I don't know if you saw on the news, like just 3 or 4 days ago, there was some picture of a guy being attacked by an octopus on the beach, which was kind of impressive. It left him with all these marks all over his head, etc. He was whipped by the arms of an octopus, an angry octopus. We'll be doing eye clicker today. Yes, if we have time. Maybe not. If not, we'll do the eye clicker questions right off the bat at the beginning of the next lecture, okay? 2 minutes, yes. Many cephalopods have chambered shells, or many of the early forms that you see in the fossils. The only sort of remaining ones that have that these days are these nautilus. There's only a handful of species that occur in the Indo-Pacific. And this cross-section of the shell, you can see it's got these chambers connected by a little hole each way, and that's used for buoyancy within these, okay? Most cephalopods retain a small internal shell. That's true for all of them except for the octopuses, which have lost the shell altogether. Cuttlefish have a large calcareous bone inside of them that people use for giving to birds in cages and things. Squid have something similar, but it's more reduced and a bit more flexible than those of a cuttlefish. Okay, yes, we've got time for a few quick eye clicker questions then, okay? So, move on to that, and I'm just going to rush this because of time. So we're going to start this, and because somebody says, kind of feel like an eye clicker today. So here we go. Okay, so I've set that going, and the first question is Lophotrochozoans, the group we've been talking about. Hopefully this is an easy question. Are plants, bacteria, fungi, protostomes, or deuterostomes? Can't see it, it's there. Okay, I have it on my phone as well, so I'm going to see if it, because I've been like, you know, making sure this works by like putting it on my thing. I can see refresh. Yeah, so maybe try refreshing if you're not getting it to work. Okay, what have we got? We've got 156 answers right now, okay? And there's typically about 220 people on the, well, there's 201 people on right now. So most people have been doing it. 167. Okay. So, right, I was able to do it. So I'm going to cut that off now, okay? No option for me. Yeah, so I can't answer. You got to remember that each one of these is worth like a tiny fraction of 1%. I mean, literally, okay? So I'm going to cut that off there. And the answer is, okay, great. Glad to see that. Most people have got it correct. It's D, okay? So we've been talking about the Lophotrochozoans, which are a type of protozoan, okay? I know we're pretty much out of time, but just going to do one more, which is the Lophotrochozoan group with the most described species is the nematodes, the mollusks, brachiopods, annelids, or the insects. Okay, let's see, what have I got there? Okay, so I've managed to answer here. Hi, Professor, I could not see the question on my iClicker, so type my response in the chat. Okay. What do you mean by described? What do I mean by described? As in, a scientist has given them a name, okay, so that they've been named by science. That's what I mean. There might be a gazillion other species that haven't been, described by a scientist yet. Yeah, the most known species, the greatest number. Yeah. Okay, I'm just looking at the questions here, which the comments. Okay, so we've got 184. I'm gonna click it there. Okay, and so great. Most people have got that correct. B is correct. We described how there's about 117,000 described mollusks, which is far more than any of the groups within the Lophotrochozoans. Insects, as we'll talk about next time, have way more species, but they are not Lophotrochozoans. Okay, they are protostomes, but not Lophotrochozoans. B, but said no. Okay, I know there's some people who had issues with the iClicker. Remember, it's literally 0.1% in total or something. Okay, great. So I will end it there, and we will continue next time and talk. Next time we'll talk about the ecdysozoans. Remember, there is no. I talked at the beginning of this class about how it's important to do the pre-class quiz. There is none for Thursday though. Okay, so don't do that. Just upsetting. Okay, great. Sorry, just reading more of the questions and the comments. Okay, good. I hope you all had a good break, and I will see you soon. Okay, see you on Thursday. And
Professor?
yes, is that Melissa or who is this?
Yeah. I just had
Okay.
a quick question. Based off of seeing like our pace today, do you think lecture 17 will be on next week's exam?
No.
Okay, thank you.
So, well, I mean, what we cover in lecture 17, yes, but it won't be the deuterostomes, okay?
Okay, thank you.
Okay. So we will only be examined on what we've covered up until that time.
So, Professor,
Okay.
Sure. Professor, probably just the second part of protozomes. That's all we're going to get to for the exam.
We'll probably do the ecdysozoans. I've got less slides for the ecdysozoans, the next group of the protozomes, but it'll include protozomes 1 protostomes 1 and 2, but not deuterostomes 1.
I got it. Thank you.
Okay. Great. Any other questions? I've got there's office hour from 2 to 3 today, in case people have personal questions, or I don't know, just anything you want to talk about. Or you can ask me something now if you want to do it in this setting. Office hours today, yes, 2 to 3. Okay. IOT. Or yacht. Sorry, I don't know how to pronounce that. Are these valid responses valid? Let me. Sorry, I'm just seeing some of the individual comments here. Could you see the question mark? I click as a type response. Okay, so let me make a comment here of. So I don't know how to pronounce your name, I'm afraid, but I will see if I can modify your iClicker thing. If it doesn't, like I said, it's like 0.1% or something. And Michael, hyperperson responses. That's a question. Okay, Michael, I'll have a quick look at yours as well, okay, and see if there's a way of figuring that out. Okay. Okay. Great. Okay, we got any more there? Yep. Great. Okay, thank you all, folks. I will speak to you next time or in office hours later if you want to do that. Okay. Bye bye.